Hello and welcome to this Australian BioCommons webinar. My name is Melissa Burke and I'm the Australian BioCommons Training and Communications Officer. I'll also be your host for today. In this series, we aim to share useful information about the latest digital techniques, data and tools for the life sciences community. Each month, we hear from our local and international peers who present a bioinformatics topic that we hope will support Australians to deliver their best environmental, agricultural, and medical research. Before we start today, we'd like to take a moment to acknowledge the traditional owners and their custodianship of the lands on which we meet today. In my case, this is the Gidabal people of the Bundjalung Nation. We pay our respects to their ancestors and to their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Today, we are thrilled to welcome Dr. Allison Heath, Dr. Jack DiGiovanna, and Associate Professor Mark Cowley to speak to us about their work towards achieving harmonised analysis of childhood cancer data across international borders. Dr. Allison Heath is the Director of Data, Technology and Innovation at the Children's Hospital of Philadelphia. She is responsible for providing technical and collaborative leadership to build platforms for data-driven discovery that span across both research and operational efforts. Allison and her team have been working on solving big data challenges in the unique context of pediatric diseases, including leading the development of the Kids First Data Resource Centre. Previously, Allison was the Director of Research at the Centre for Data-Intensive Science at the University of Chicago, where she led several large-scale data-intensive projects, including the NCI Genomic Data Commons, the Open Science Data Cloud, and the BioNimbus Protected Data Cloud. Allison completed a Bachelor of Science, a Master of Science, and a PhD degree programs in computer science at Rice University in Houston, Texas. She is committed to accelerating discovery and translational research by democratizing data access and analysis. Dr. Jack DiGiovanna is the Program Director at Seven Bridges, where he supports the programs team, determines overall strategy, engages with prospective clients, and sets product development priorities. While at Seven Bridges, Jack has also served as the General Manager for Diagnostic and Clinical and the Programs Team Lead. As the Programs Team Lead, he contributed to multiple projects, including the API and the development of the Seven Bridges Sonar, a tool for interactive exploration, cohort selection, and hypothesis testing. Prior to joining Seven Bridges, Jack was a postdoc and then a group leader at ETH Zurich and École Polytechnique Fédérale de Lausanne in neuroscience, machine learning, and prosthetics. He earned a Bachelor of Science in Electrical Engineering from Penn State and a PhD in Biomedical Engineering from the University of Florida. Associate Professor Mark Cowley is a computational biologist whose expertise is in genomics, precision medicine. Mark joined the Children's Cancer Institute in 2018 as the head of computational biology, the co-head of the Luminesce Alliance Childhood Cancer Computational Biology Program, and is now also the co-head of the ACRF Child Cancer Liquid Biopsy Program and a conjoint associate professor at UNSW Medicine. Mark's multidisciplinary group develops novel analytical approaches to understand the molecular basis of disease, usually from patients enrolled into precision medicine studies. His team also develops the digital infrastructure that underpins the Zero Childhood Cancer Program, an ambitious national research program that uses precision medicine to diagnose and treat Australian children with cancer. Welcome, Allison, Jack, and Mark. I'll now hand over to Allison to get started with the presentation.
Great, thank you so much, Melissa, for that kind introduction. I'm just gonna share my screen here. And really excited to be here today with Jack and Mark to talk about, I think, some really exciting work that we've been doing over the last year. You know, it's funny, I was reflecting on, I think a lot of this started with conversations going to some of the Lauren conferences and tossing ideas about, and it's been really great to see the Australian BioCommons emerge and really provide us a practical framework to do some of this work. So I'll just start out a little bit with kind of the idea of what is a data commons from, maybe my perspective, having worked on this, really starting from cloud computing and big data, really driving some of the different technologies. I found this old slide I used to present back in 2015 of thinking about new models of data, moving from a model where people are trying to download it locally in labs and there's many copies of the data, to really having what has kind of emerged as a commons like infrastructure where you can bring compute to data, have different virtual infrastructure. And what's really been exciting about this project is what are the technical steps from that to really start supporting more precision medicine, more real-time things across the globe. So I think when a lot of this originally started in the, kind of 2015, 2016, it was really focused on how do we collate data, the computing infrastructure, some of the core services and tools. And it was really around some of these, big projects such as the Cancer Genome Atlas, other large scale data infrastructure, but we're starting to realize that data is being generated all across the world, and especially in pediatrics where we have a lot of these rare diseases that we'll talk about one as a specific use case, to how do we leverage all this technical work to get to a point where we can really start doing some interesting science and even ultimately impacting clinical care. And from that, there was a lot of different people doing this. I think there's this really nice Medium article about talking about the data biosphere, and really came out with principles of how do we do this modular, community-driven, open standards-based. And GA4GH, the Global Alliance for Genomics and Health, has really been it's just been really great to see that emerge as a great community forum that a lot of this is coming out of. And then really during this project, thinking about how do we apply that more specifically to some of the challenges in pediatrics, specifically pediatric cancer. So as Melissa mentioned, I've been involved in the Kids First Data Resource Center, which is a project out of NIH, really focusing on looking at doing whole genome sequencing to really understand childhood cancer and structural birth defects. And here's the mission statement of the Kids First program, and I really highlighted the collaborative efforts, which this project has really highlighted some of the power we can have if we really collaborate on some of these data sets. And it's been a really unique experience trying to intersect many different data types and really taking a lot of that original technology, the cloud computing, the different data commons, and applying it to a very diverse set of data across different organs, across different disease types. And so over the last couple of years, Kids First has generated a lot of data. We have over 1 petabyte released right now. We have 13,000 participants and quite a few more away. So over 21,000 more funded. And over the next couple of years, all this data is being rapidly released. We really try to adhere by the NIH genomic data sharing policy, which says 6 months basically is embargo time, at which case it comes available to the public. And we've even been piloting with some of the other data sets of making it even more rapidly available than that. And so in order to do this, we really had to build a framework that I like to talk about interoperability. So a kind of ecosystem of different tools, including a portal, integrating with other portals like CBio portal, thinking about other data services, and again, leveraging a lot of these standards through things like Gen 3 and FHIR. But today we're really going to talk a lot about what we did with Cavatica and how using that cloud-based workspace and this data commons like capability, we can really facilitate international collaboration. So when we started this project, we really tried to nail down a key use case. And one of the data sets that's supported by Kids First and that we help operate at CHOP is called the Children's Brain Tumor Network data. And so in the US, there's about 4,000 children diagnosed with a brain tumor every year. The median age of death is only 9, and you can see the survival rates are very dismal. And we have over 14,000 children living with a brain tumor every year. But if you compare this to common diseases or other data sets, there's actually, while there's a fair bit of information here, there's a real existential need to combine data sets across the world. And one of the things that the Brain Tumor Network and PNOC, which is the clinical trial operations that we have a very close partnership with, is to identify sites around the world. But, one organization isn't going to do this all. And, Mark's going to talk a lot about zero childhood cancer here in Australia, which I think is a very impressive effort. And so really looking at the brain tumor data, we thought this would be a great use case to drive the technology, the engineering challenges that we need to address to be able to co-analyze some of this data and make it available as rapidly as possible to each other. So this is just a dashboard of some of the data sets that are available today that we are using as some of the driving use case. There's a wide variety of different brain tumors, some of them very rare. So, even though I gave aggregate stats, looking at the different histologies, there's a lot of, I think, interesting things that we can do as we combine those. And this is just a shot of where I hope, as we come up with these commons, we can start going where we can use the data sets in the commons as a backdrop for as we get new patients, as we enroll new folks in clinical trials. This is an example of one of our molecular tumor board reports that is trying to show a landscape of where does that new one lie in this genomic analysis. And I think towards the end of the presentation, Mark will show some initial steps towards this. But keeping that as the vision, there's a lot of key engineering that has to happen to make that endpoint. And so this project really was trying to identify those, pilot those with real data, with real use cases, and kind of come up with what does this infrastructure look like? What's how do we move from kind of a national infrastructure to an international one? And so as we develop this project, you'll hear Jack and Mark talk through a lot of what was accomplished in terms of creating workspaces in Sydney, optimizing some of the workflows, doing the different processing in the different regions of the different data sets, and then bringing it together. And then really also thinking about how do we go from pure cloud approaches to hybrid approaches where we have integrations with local infrastructure, and really trying to optimize how we do that. So with that, I will hand it over to Jack to talk about how we did Cavatica Australia and some of the more in-depth engineering that goes on.
Thanks so much, Allison, and thanks, Melissa, also for the introduction. Just bringing up the screen. Sorry for the delay. So thanks so much. I wanted to quickly introduce Seven Bridges for people who weren't familiar. We founded in 2009. We're a leading cloud-based collaborative ecosystem provider. We work with both commercial, like biopharma, but also NIH cloud systems, which is what our team is responsible for. Working with the NCI, with NHLBI, working with Common Fund to co-develop Cavatica with CHOP. We have a large engineering staff. There's about 300 of us, but there's also a large percentage of bioinformaticians with about 50 of them on staff. Really focus on user-centric design and fair data and actually fair workflows as well, which has gotten us deeply involved in GA4GH data and analysis standards development, and have a good understanding of cloud security and cost optimizations. I joined the company about 5 years ago, and I was looking through old slides and pulled up our marketing materials from then. And what I really liked is that a lot of people at this time with Seven Bridges was really pushing this idea of a single pane of glass. So in a case, what if the data was here and here, how would you analyze it? What if you had researchers here and here? So some of the things that we built to enable that are actually wound up being key to this collaboration with the BioCommons, with CHOP, with Zero. And those two things are really connected cloud storage and multi-cloud, which we launched in 2018. And while in 2016, this was a nice marketing piece, what if we had the situation with data? I think more and more we're realizing that we are in the situation where there's either policy level restrictions or geographic restrictions where it's just not feasible to keep moving data around. As Allison introduced, it was a big deal to get the data into the cloud and not just keep downloading it, but there's more than one cloud. So how do you manage that and how do you move through that? And that's what I wanted to talk through in this presentation. So we started working, I think it was about a year ago we started. We've been talking for a while with Zero and ARDC and BioCommons. Really great collaborators. They have all of their data here, either on AWS Sydney or the National Compute Infrastructure. And then Seven Bridges and CHOP co-developed Cavatica in 2016, initially for pediatric cancer, and then now under the umbrella of Gabriella Miller Kids First, also for structural birth defects. with most of the data here in AWS US East 1, also capabilities in Google West 1. So the challenge, as Allison nicely introduced, was you need, because this is a rare disease, pediatric cancer, as a new patient comes in, it would be very helpful to be able to aggregate the data from other sources around the world. But what you don't want to happen is a new sample comes in, you ship it over to the US, you process it, egress it back, or vice versa. So that's kind of the technical challenge, that you don't want to be moving data around. The scientific challenge we focused on in the last year, which Mark is going to get into much more, was if we could share and combine this brain cancer data, how would this help to discover new disease subtypes? How would you be able to look, as Allison showed in the last report, as a new sample comes in, where's that patient cluster with existing patients? So really exciting for both from a technical and a scientific perspective. So the two data sets that we used here are coming from CBTM. There were about 900 RNA-seq samples. These had all been already processed on Cavatica with the Kids First RNA-seq pipelines. Those pipelines are available on Cavatica or on GitHub as well, if anyone is interested. They also had whole genome sequencing data from about 8,000 patients available. And on the zero side, there were 112 samples, get my pointer going. There were 112 samples available in NCI Australia with both transcriptomics and whole genome seq data available. So the overall processing flow that we were looking at here in this flagship was, for the transcriptomics data from zero, process all of that uniformly with the Kids First RNA-seq workflow to get to gene expressions. For CBTN, this has already been processed, so we have gene expressions ready to go. And then do batch correction and clustering, and you could, with the aim of identifying distinct brain cancer subtypes, perhaps novel subtypes as well. Once you had that information, you could further take the whole genome seq data, put that through zero structural variant caller, and try to understand shared molecular drivers for patients who show up in these distinct subtypes. Now, in terms of processing, we're going to be sending the processing to the data. So operating in different areas, and reproducibility is quite important. This may be very familiar for folks in the audience, but in case it's not, a quick overview of how we handle this reproducibility is with containerized code and a workflow description language. So if you look at the spectrum here from publication or development to reproducibility or a production level flow, the minimum you need is just the code. But if you use something like Docker, you can now containerize the environment that you are working in, so you're sure wherever you load it, you're coming from the same starting position. Common workflow language is wrapping everything together, encapsulating the requirements, how the code relates to the Docker images. There's other workflow languages that do this as well. Examples are WDL, Nextflow. We use CWL here. But if you have all three of these things, you're able to really have the complete package. And as long as you move it to somewhere that has executability and the data exists, you're able to reproduce the exact same workflow in both places. In terms of architecture, Cavatica is built on the 7 Bridges core infrastructure. So here I'm showing all of our different microservices, abstracted out the names. They're all very cool names and be exciting to dig into, but you can imagine this is doing the UI, the billing, admin, task service, file service, viz, different things. It allows for diverse users to come to the platform, potentially through GUI, through API, through Shiny apps, but they're all working with one platform. And one of the first thing I talked about that we built in that overview slide some years ago, I think actually we developed this while we were developing Cavatica with CHOP, was the ability to connect to other cloud storage. So this would be a bucket that's not owned by 7 Bridges, but is owned by one of these users. And they could access and process their files just as easily as if it were on our buckets. So the first thing we did in this project is to add a connection to the NCI Australia buckets, or we're actually developing this as we go right now, and it's in final testing, such that if there was data locally on this HPC, it could be visible to the platform. We could use the executor, we could use the workflows, we could use all of the processing capabilities, streaming this data in just on demand as it's needed. So I put together a quick video of what it looks like to work with data that's on one of these volumes. Just before the end of the year, Broad put Nomad on open data from AWS. So what we did here is the user already connected to that volume. And now in the project, they're going to add files. Instead of using an uploader or pulling from a data set, they select the volume, and here they're just traversing the directory structure of those files. They're going to go select a VCF and an index file and import them to the project, which is just going to create a pointer within their project to those files. What's nice though is these files are, first class citizens. You could associate metadata with them. You could process them with tasks. You could download them. You could put them into RStudio, whatever you like. So it's really making the files available and putting them in one UI to interact with. So this solves part of the problem of connecting to different clouds, but it only solves the problem very well if the cloud is in the same region as where you're going to do the processing. So the prior solutions, if you were on a 7bridges platform prior to 2018 or some other solutions, they would bring data to the compute. So let's start with the transcriptomics data in Sydney. You could create a project and analyze this in Sydney, and that would be fine. There would be no egress. That would work really well. But as soon as you wanted to bring over the children's brain tumor network data, you get this scary red line because this is egress. You're bringing all the data over before you process it, which is really not ideal. Here we introduce multi-cloud for the part of the diagram I didn't explain. Here are projects on 7bridges. So we've always supported the ability to store and compute on US East 1 for Amazon, US West 1 for Google Cloud. And what we did in this project is add capabilities to also create projects, store and compute in Sydney for AWS. So now we have a situation where these data sets are in the same place and we can avoid egress for processing. Projects, I just want to give a high-level overview. You could call them sandboxes, workspaces. There's a number of different names, but for us, they're the base level of organization on 7bridges platforms. You have a project owner. She can add project members as she wants, assign them role-based access control, and all files or pointers to files, apps, which are tools, workflows, pipelines, R scripts, or anything else, and tasks, which are the execution of the apps, live in that project, and any files that are generated will also live in that project. So they're really important for collaboration and interoperability. It also gets important when you start to think about controlled data access, making sure that you only invite people. It makes a nice container for only inviting people who have access to that data. And here's an example of me creating a project in AWS Sydney. So I'm quickly just typing a name. There's going to be a drop down right here that I select, hit OK, and with under 10 seconds, I now have this project organized in Sydney. And that allows me to bring, with some foresight on the user side, it allows them to bring compute to the data. So we're in the same situation. We have the RNA-seq data from zero, create a project in Sydney, process it there. That works just fine. For the children's brain tumor network data, we'll create a project in US East 1, processes there. We avoid egress. We're using CWL and we're passing that workflow, so we're sure we have exactly the same result. And this might be the end of the story if it's something you could do with federated compute. If there's a situation, in this situation, there's no free lunch, there will have to be a little bit of egress because we need to combine the gene expression results from both. So you do get a little bit of egress here, but it's orders of magnitude smaller files than if you would have brought over the raw FASTQs. And depending on the type of data you're working with, I mean, this can be really huge and this can be very small. And in the end, it's going to be from a project in Sydney using a Jupyter notebook for integration and batch correction and unsupervised clustering in RStudio. And this is the point where I want to hand over to Mark, who's going to get into more of those details.
Welcome everybody online. It's my pleasure to be here today and tell you a little bit more about some of the scientific drivers and underpinnings of this project. I'm presenting on behalf of some of the members of the computational biology team at Children's Cancer Institutes, especially Chelsea, Marie, Sabrina and Louise, and generally on behalf of the Zero Childhood Cancer Program. All right, so 2 or 3 slides on what Zero is, and then we'll get onto the Australian BioCommons project. So Zero Childhood Cancer is Australia's national precision cancer medicine, pediatric cancer precision medicine program. So essentially, we're about 5 years through this program. We went through a target phase based in Sydney, now a national phase, and we've recruited about 450 Australian children to the program, all of which have very high risk cancers, poor outcome cancers, those children that have less than 30% chance of surviving 5 years. Over on the right, what I'm showing is that we perform one of the most comprehensive molecular profiling programs in the world. So deep whole genome sequencing of the germline and the matched tumor, tumor RNA sequencing, methylation profiling, and where possible, we take pieces of tumors and subject them to high throughput drug screening, inoculate them into PDX mouse models. And ultimately, the goal is very much clinical, to impact clinical care and improve outcomes for these children. We're near the end of this second phase of the program and just received funding to expand the program nationally, ultimately to every single child or young person with cancer in the country. So that becomes about 1,000 patients a year being eligible for this program at all 8 sites across the country that treat these types of children. We live and breathe precision medicine, but I think this slide really captures it for those that are not deeply involved in precision medicine. The idea really is that you have a child present at hospital, in this case with a very hard to diagnose tumor, failed the standard pathology tests, and this child was about 1 year old at the time, and the tumor was filling up her chest cavity. So things were pretty dire for this little girl, and this is about the fastest we've ever been able to do it. I'm not going to pretend we do it this quick every time, but essentially in 9 days from that patient being identified in the hospital to being sequenced with our partners at the Garvan Institute, analysed on our existing commercial cloud platform, which in this case is DNA Nexus, analysed, processed, we identified a novel form of a really important driver fusion, a mutation involving the NTRK3 gene. And that was remarkable because about a month or 2 before, a novel targeted therapy had been developed specifically for this type of gene fusion. And so we were the first in the country to get access to this drug and improve outcomes really rapidly in this patient from this program. So a couple of themes in there. Drug development is happening really fast in parallel with the technological developments, and that when you get to match that patient to the right treatment, occasionally you get absolutely remarkable outcomes. And we're able to make those recommendations back to the treating oncologist in near real time. So 2 years later, this child's doing brilliantly from this. So that is kind of precision medicine in action. This is not just one example. So this slide is meant to convince you that when we've done this and evaluated the program over the first 250 or so patients, that in almost all patients, the molecular profiling strategy informs the diagnosis of that patient. So 94% of the time we find a genetic change that we think is clinically important. Remarkably, in over 70% of patients, they get a targeted treatment recommendation. This is much higher than we thought when we set out 5 years ago along this program. 16% of patients had an inherited genetic mutation, which really impacts the risk of further childhood cancer in that patient's family, and maybe explains why those patients develop cancer in the first place. Really importantly for management is getting the diagnosis right. And in 5% of the time, we change the diagnosis. And so really, that's really cool. That's great. But what impact does this have on patients? In 70% of patients that took that targeted treatment recommendation, there was an objective clinical response, either a tumor slowing, shrinking, or achieving a stabilization of their disease. So that's kind of the slides to convince you that this is an effective strategy. But now I want to switch gears back to, I think that also justifies why Zero is sort of being included in the Australian Bioinformatics Data Commons, this pilot program. And I'm going to focus a little bit more on the data challenges that have been alluded to throughout. I kind of mentioned that Zero is a national program already. Every single pediatric hospital, of which there are 8, are recruiting for the study. There are dozens of additional research partnerships, a number of international partnerships. And so no longer can we, not that we ever wanted to, but no longer can we kind of hoard the data. Our objective is to share that data with our partners nationally and internationally as straightforwardly as possible. But those of you that have shared large genomic data, you know, each data from these patients is about 1 terabyte. Sharing that data is kind of tricky. So this is kind of the objective, particularly for this year and beyond, is how do we share the data so that we can accelerate research and improve outcomes long term. And that's why there's a perfect sort of synergy with the goals of of the Kids First team. So the three main challenges I'd like to talk a little bit more about is sort of at the start of this project. We were in a scenario where we were already using the cloud and had been for many years, developed nearly 140 analysis apps, but they were locked to that single cloud vendor. So it was increasingly over that time, as has been hinted at today, the idea that multi-cloud, you should be able to run your tools any way you like, and wherever that data is, you should be able to run your tools. So that was a limitation of our current setup. The pipelines were becoming increasingly complex, increasingly costly. Flexibility was becoming a challenge for us as we had lots of different scenarios, you know, cell lines with tumor only, PDX mice where you want to subtract out the mouse component, and even some children who have had stem cell transplants. And so they're a 3 genome problem because they have their own germline, their own tumor, and a donor genome floating around. So the workflows to analyze this data got increasingly complex. And as I mentioned, they were kind of locked into the single cloud-based platform. Really hard to share these pipelines with people and allow them to benefit from all the engineering we've been doing for so many years. And then the last bit I'll talk about is that we now have about 450 patients. The data is about 1 terabyte for each patient. This is pretty hard to move around, and we'd really like to improve that aspect of the program. So if I start with the apps, when we started working with the Oz BioCommons and Cavatica teams, what we could find is that we were able to immediately borrow about 20 existing tools that were already Dockerized that we could just bring into our pipeline. We had to develop about 25 unique apps that we Dockerized. And as Jack's alluded to, that involves making sure all the dependencies, the exact dependencies, and anyone who's handled R being updated every month knows that the pain involved in doing that. Get the apps and the dependencies right in that container so it will run every time. And we've built 3 new tools that are specific for the precision medicine analysis workflow. The first you may have heard of, VCF Anno from Aaron Quinlan's group, but a couple of others that help get the data into our database. So VCF to TSV and our custom Helium algorithm, which tries to bring the pathogenic variants and pull them out of the large list of variants and make it more obvious that they're the ones that are clinically relevant. So our expert curation team can focus on those. This pipeline is available through Cavatica, although we are going through code review and making sure that the CHOP team agrees that this pipeline is working well in their hands. And for those that are in cancer genomics, I'll put the names of some of the tools that are on the right here. I think BWMM, GATK, and Strelka will come as no surprise to many cancer genomicists on the line. Maybe what we're doing a little bit differently is working closely with the Hartwig Medical Foundation, who have developed a suite of tools for really accurate copy number estimation. That's the amber, cobalt, purple tools, and GRIDS2 from also a collaboration with Hartwig and Tony Pappenfuss's group at Epidemic, a really great but resource-hungry structural variant caller. So that's kind of the apps. Then we've got to bring them all together into workflows. I'm focusing here on the DNA workflows, even though I'm going to tell you about the RNA-seq sciences, because this was the bit that was quite unique to our workflow. So you can see here the green dots are each step. And like most workflows, they have lots of inputs. Outputs from one step are passed through automatically as inputs to the next step. And so part of the benefit of doing this and Dockerizing everything is, and actually using the cloud in general, is that some jobs are not very resource hungry and you can have them run on a small, cheap VM, like a single core VM with low memory. Others, like grids, are resource hungry and you need to run them on much larger VMs. So when you get to build these things and optimize them, you end up being overall much more efficient because you're not stuck with the instances that your HPC can provide you. You can pick and choose the instances that you want. Ultimately, this leads to a more efficient workflow, optimizes cost. And a couple of the reasons that Jack really focused on, which was attractive about the platform, is you're not locked into storing your data within that platform. You can connect it to potentially to your data at the HPC or your data that's on-prem or a different bucket. And this was really attractive for us. The Dockerization gives you that plug and play capability. And this gave us the opportunity to add some features to our pipeline, like CRAM support. We're just finalizing the testing of that, but it's looking really good. And upgrading our apps so that every one of them is Build 38 aware, the latest version of the human reference genome. When we've built new apps, we've also tested that they're multi-cloud compatible with the previous apps on our previous cloud-based platform. So that was a really big win for us, because I really don't want a situation where we've got a pipeline at NCI, a pipeline on DNA Nexus, a pipeline on Cavatica. I want to see this all harmonized so that a single engineering effort can then, and a single maintenance effort can scale across all those different platforms. So that was a big win for us. We're calling this pipeline Graphene. So we managed to get a name out late last year for it. And besides all those, those great achievements that I've just told you about, the multi-cloud, the cost, the optimization, it also ended up saving us time in each analysis job and saving us cost on each analysis job. So this is an incomplete slide, but the timings I could pull together this week were on most of our post-alignment steps, except for grids. And grids is the slowest. I will add that. But overall, what we're seeing here is a 13% reduction in the time it takes to analyze the equivalent data on the two platforms, and a nearly 50% reduction in costs, which is obviously, as we start to process more and more data in the cloud, that's a really important thing to us. It's also important, it's important for each new patient going forward, but it's really important when you want to start to think, how do we scale out our analysis to thousands of data sets that exist around the world with our tools? Cost becomes a real, because you can kind of budget in the cost of each patient into the program. But if you're going to do exploratory analysis on thousands of patients, then the costs really stack up. So this is really promising. And yeah, it's been going really well. So this is the scientific part of the presentation. So back to that question that's been alluded to a few times is, can we share, combine and integrate brain cancer data from local, zero and international patients, and ultimately hope to discover new disease subtypes? I'm here to say that we've spent about a week working on this, and I don't think we have discovered a new disease subtype yet. So if you tuned in to hear what our new brain cancer subtype is, I'm sorry I'm going to disappoint you. We are going to keep working on that. Just to reiterate what Jack has mentioned, is that the way we actually did this was to upload Zero's data in NCI Sydney into the Cavatica workspace in Amazon Sydney. From there, we've imported 1,000 brain tumor results already processed from Kids First, the pipeline in Cavatica. Harmonizing the diagnoses was actually quite tricky, because whilst the brain cancer patients, we diagnose them differently in the two countries. So in this phase of the program, we had fewer patients, so we tried to re-diagnose ours to match the diagnoses from CHOP. But this is definitely an ongoing effort and a goal that I'm going to talk to about 2021's goals. Is how we do that more systematically. Clinical metadata sharing is quite hard. With the methods, we ran the RNA-seq pipeline on the Zero data, loaded those results into a Jupyter notebook for QC, batch correction, and then into RStudio for clustering. Again, with that goal of identifying distinct cancer subtypes, ideally with patients from multiple geographical regions. So the reason I spoke to you mostly about the DNA sequencing pipeline is that we had some custom parts to it. Our RNA sequencing pipelines actually were quite similar with the CHOP group. So we've adopted the Kids First RNA-seq pipeline. The link to GitHub is there. It's easy to find, and it's easily available through Cavatica. Many of you in this space will know these tools quite well: STAR, RCM, and Callisto, Ariba, and STAR Fusion. And recently, the CHOP group has developed AnnoFuse to help you merge the results of multiple fusion calling algorithms and harmonize that data to make it more comparable. As Jack's already shown you, the egress was quite small because we're pulling in processed data from America rather than the raw data. And we brought that into the Amazon Sydney workspace with Jupyter and RStudio. So it was a really quite an efficient process to suddenly obtained nearly 1,000 brain cancer patients. So I need to shout out to Chelsea from our team and Jolyn from the CHOP team who've been working hard on this all week. On the left, I'm showing you a T-SNE clustering of the RNA sequencing data combined between 0 in red and blue or green from CHOP. We've worked really hard as much as we can to do some batch correction, but I think you can see that green cluster still stands out, and maybe that suggests that there's a little bit more batch correction that we're going to need to do to truly harmonize and analyze these data together. And then when we switch on the right-hand side to starting to label the patients by their diagnosis, at this stage, we're seeing a little bit more scattering of those clusterings than we were expecting. And this to me suggests that we need to do much more investigation on optimizing the feature selection here so that we can get nice distinct clustering of different brain types, brain tumor subtypes, to then start to dig into clusters of them that have potentially shared molecular drivers. So this was hoping to be the figure that we just popped out with a quick analysis in the last week, and this is the one we're going to keep working on a lot. I'm still optimistic that there are going to be clusters of Australian and US patients with unique molecular phenotypes, but that's what we're going to keep working on. I've got to, and I realize we're at 44 minutes past, so I'm going to just end on a couple of ideas about where to next for the Oz BioCommons program. And really, it speaks to the third challenge that we had, which is the challenge of data sharing. We've only got 450 patients right now, but on the left, you see that the amount of storage ramps up pretty quickly as we start to expand our program. We'll be at 2.5 petabytes before we know it. CHOP's already at 1 petabyte, so I'm sure they'll be way out in front again. But for us, that's quite a considerable challenge, is how do we share that data? We just published that paper that I showed you at the start of my presentation, and it's still taken us 6.5 months to get these 250 patients of data into EGA. And that's not through lack of trying. It's been a really hard process. So data sharing is hard. As I've said, we want to collaborate and make this data shareable. And so really, this is one of the main goals of the Oz BioCommons Human Genome Initiative Project 1 for 2021. So this is the technical goals of this program, which is really to try and make data sets much more fair, findable, accessible, interoperable and reusable, and where possible, use emerging open standards, such as those from Global Alliance. One of the things I'm most excited about, being a bit less of an engineer here and more of an end use case kind of guy, is the building a Gen 3 powered data commons platform so that people are going to be able to start to see what data is available, the clinical metadata that's available, and have that linked to the underlying genomic data. I should add that this program is now beyond just zero. It's going to include the University of Melbourne Cancer Research Program, and Oliver Hoffman's heavily involved in that collaboration. Once we have the data commons dashboards, we'll have a data migration program, training and documentation, so that this is extensible to beyond these two groups we've just mentioned. And as I've said, where possible, using open standards so that people can authenticate themselves with passports from multiple different authentication providers, using Global Alliance APIs like the Data Repository Service to share data from wherever it lives, increasing use of the Common Workflow Language, Tool Registry Service, and the Workflow Execution Service. So a whole bunch of open standards for enabling reproducible research on data globally. The bottom line is about an Australian service for integrated identity and access management through CI Logon, which we're going to be trialing. So that's the big focus, is on making the data much more shareable. There's some scientific goals that help underpin these engineering goals. And briefly, these 5 activities are to build a national pediatric cancer genome data commons platform so that we can share this data much more easily, to focus not just on the RNA sequencing analysis of Australian and American patients, but to increase that onto the structural variant landscape. We've mentioned grids before. We think that's a great structural variant caller, and we're looking forward to running that on a really large scale data set and learning more about brain cancer structural variation patterns. We'd like to develop a harmonized pediatric cancer clinical metadata dictionary so that we're calling our patients by the same diagnoses, or at least we can interoperate that data. We'd like to build a pediatric cancer knowledge bank so that our experience in Australia can be shared with CHOP and vice versa and others. And ultimately, this is a stretch goal, I guess, is can we start to think about accelerating precision medicine through sharing molecular tumor board findings? So what I mean by that is if an Australian patient this week had a certain mutation and we made a drug recommendation, how might the CHOP team use that information in their molecular tumor board next week? So that's a stretch goal and really where we want to get to. So the take-home messages from today's presentation from the three of us really is that the Cavatica platform is now available in AWS Sydney. I've shown you the cost and price impact on our already heavily optimized pipeline. Through multi-cloud and connected cloud storage, you can start to think about a single pane of glass to analyze data that's geographically distributed. This is increasingly important today. We've been able to create this strategic partnership to aggregate the largest data sets of brain cancer in Australia. And we're still working on the research outcomes of that partnership. Our pediatric cancer whole genome analysis pipelines are now available, or soon available. Contact me if you're interested. And as I've reiterated, the 2021 goals are to sort of focus on data sharing, building that Gen 3 powered BioCommons web interface that looks a bit like the Kids First data resource portal, and focus on authentication, security, sharing, and making that data accessible. So it's my pleasure to acknowledge a really fruitful collaboration with a large number of people at many different sites. Because they haven't had a chance to say anything today, we really want to shout out to the Australian BioCommons team, especially the leadership of Andrew Loney, Stephen Manos, Paul Coddington from ARDC, and from NCI, Brian Davis, Daniel Rodwell, and the folks from ARNet Bioplatforms. So with that, and of course the members of our respective teams that we've represented here today. So I think with that, I will stop and look forward to taking any questions from the audience.
Thanks very much, Allison, Jack and Mark. We do have time for questions. Please do write those into the Q&A panel that you see on your screen, and our speakers can see the questions you're writing in there. I can see there are a few questions have come through already. So when you're ready to answer them, you can read them out and answer them verbally.
So I can combine the first two, I think, and Allison, maybe you want to join in on the answer. It's interesting to know the Pathfinder is incorporating the testing of GA4-GH standards. Is there a plan to do this? It seems like a public-private partnership would be a great way to do that. And totally agree, as we mentioned on the slides in a few different places, for Cavatica, we were one of the first, if not the first, platform to stand up a DIRS server for controlled data. So that is active on Cavatica. We're also active in the FASP workflow and demos there to bring in WES and TERS standards. We've been working, our team, the CHOP team as well, with the RAS, which is being able to incorporate the GA4-GH passports for authentication, which is, I think, as Allison and Mark both pointed to, a very hairy problem in this case when you're starting to combine the data from different places. And Allison, I don't know if you want to talk about FHIR as well, which is coming online.
Yeah, so I mean, I think this project really, we focus on a lot of the genomic data, and you know, I think these questions were answered before we had those last couple slides where Mark really, I think, articulated that part of this initial part was really realizing we should align with those standards, and I think that's where some of the Gen 3 technology comes in as well. The clinical data kind of problem, which we didn't address as much in here, but I think Mark alluded to, with some of the diagnoses and, you know, ultimately for precision medicine, what are the outcomes, what are other adverse events, and how we share and interchange that is going to be really important. I think some of that is kind of coming up with the right terminology, the different subtypes, how do we encode those? And then the Kids First team has really been working on some of the NIH's FHIR working group, where we've been really like exploring those APIs for interchange, you know, because there's one world where you can kind of create a matrix or a clinical file and try to exchange that. But introducing APIs, just like on the genomic side, I think really allows for an ecosystem of different applications, different tooling, which might end up being a matrix, just like, you know, our CRAM and BAM files end up being a matrix of gene expression. You can imagine that, but thinking about how some of those APIs work, I think are key. And maybe I'll lead into the other question of how is the 7 Bridges technology different or complementary to the Gen 3 technology? So I flashed up a slide real quick there where Kids First, we actually use Gen 3 as, I really think of it as kind of our governance setup. So how do we do authorization? How do we do dearths? How do we make sure that data is distributed? And really in the US, the model has been actually different institutes in NIH actually have their own Gen 3 instances. So Kids First has one, Heart, Lung, and Blood has one, National Cancer Institute has one. And it allows us the governance, the ability to share, the ability to manage that with the standards emerging from GA4GH. And there are capabilities of, you know, having Jupyter notebooks and things like that, but really the power of 7 Bridges has been, you can plug into any one of those Gen 3 instances, have these collaborative workspace that cross multiple ones of them, and do this kind of collaborative work. And I think as Mark was alluding to, you know, ultimately we don't want competition to be about who can trap your data, but rather who can provide the best features and functionality over standards. And I think that's been a really great partnership with 7 Bridges. And, you know, I think the more we can have these standards and, you know, be able to allow it to connect in different platforms, whether they're local, whether they're in the cloud, that's really where that's going. But that's kind of at least how we currently use Gen 3 versus Cavatica in the Kids First world.
Thanks for those answers. Are there any other questions? I'll give you a moment to write some things in there. While you're doing that, I have a question, if that's okay. I was wondering if there are plans to reach out to other countries in addition to the US and Australia and to bring the data into a really, really international platform and what the challenges with that might be.
Yeah, I think as we alluded to, I mean, one of the great things about working with Zero, as Mark shows, you know, they've been doing this for 5 years and I think really have great workflows. Actually, another project that we're involved in is one in the EU called IPC, which is a Horizon 2020 project. And I think been really talking to them about how we share some of this data. I think, you know, there's always challenges with federation and governance and the different systems. And so I think for us, we've really found the right way to bootstrap this is to find these key scientific drivers. And so, you know, some of the next might be really figuring out other places with the different brain tumor data that we could bring in or other close partners. But I think you're absolutely right that this is just a first step. I'm not sure, Mark or Jack, if you have more on that point.
I think you covered it extremely well, Allison. And I think, I don't know if everybody agrees, but from my perspective, the technology is there. It's really the policy space that is so fraught with difficulties and misunderstandings, and just getting the different governing bodies to align would take much longer than actually standing up whatever tech is required to connect the dots. At least that's been our experience in multiple projects, not focusing on this project, but even projects within the US where they are just two government agencies talking to each other. There's still policy things that need to be figured out there, that because the technology wasn't there before, this is not a problem that people have faced. It's a good situation that we're in, but it's a hard situation.
Yeah, I think I would echo all of those views. Again, as a sort of end user analyst type role in this program, anyone who's tried to look at dbGaP or EGA and find your favorite publications or chain of publications and go, all right, these are the 6 data sets I'd like to bring together. It's really off-putting to say that's probably 3 to 6 months of data access requests, harmonization, convincing your own local ethics board that you're doing the right thing. I mean, a little anecdote is that when we had to convince our ethics board, they wanted to see the consent, initially, they wanted to see the consent of every participant I was proposing to access. And so there's a bit of a learning process there to convince the ethics boards that the data has been shared the right way, that we're appropriate to analyze them. And so once you've got your data access now under a new Gen 3 type model, it's so much easier to bring that data into a workspace that it makes answering some questions that were just too hard before now a reality. So I'm excited to, I feel like we're at the start of this journey, chopped way ahead of us, but I feel like we're starting to turn this corner that we can answer these much larger questions on aggregated data in a way that we couldn't dream of before. So I'm optimistic about that and what role Zero can play in being one of those data sets that helps others accelerate their research.
Thank you. That sounds really hopeful, both for pediatric cancer and for all sorts of other diseases. So I think we can benefit from this kind of model. There aren't any other questions in the Q&A box at the moment, and we are coming up to the hour. So I think we will leave it there for today. Just have a couple of things to tell you about before you leave for today. So I'm just going to take back the screen sharing. So a quick reminder that this webinar is part of a series, and as we were speaking, the next webinar has been confirmed. And in that webinar, we'll be hearing from Dr. Claire Sloggett about the AusTracker platform, which is being used to track COVID outbreaks here in Australia. So that will be on the 3rd of February, and the details about that webinar will be up on our website soon. You can also find details of upcoming workshops like this RNA-seq workshop on our website too. Thanks again for joining us, and thanks to Allison, Jack and Mark for a wonderful webinar. And finally, just to acknowledge that the Australian BioCommons is enabled by ANCRIS via BioPlatforms Australia funding. Thanks very much, and we hope to see you in another webinar again soon.